morning to all of you again, and in this videos I am going to discuss about other antineoplastic agents like antimetabolites, natural products, hormone or miscellaneous agents. In earlier videos I have covered alkylating agents and platin compounds. So first is third one is antimetabolites. Antimetabolites, as name indicates, they are interfere with the metabolisms, and under this categories, the various drugs are pyrimidine analogues, and in pyrimidine analogues, drugs are 5-fluorouracils, cytarabine, gemcitabine. These are the drugs. Another drug is under this category is purine analogues. Purine analogues drugs are 6-mercaptopurine, 6-thioguanine, 6-thioguanines. There is another drugs are folic acid analogues, and in folic acid analogues drug is methotrexate. Another drug is pemetrexate. These are the drugs under antimetabolites, and how they are showing their action. Again, problems with these drugs are their ADRs, and ADRs are bone marrow suppression, mucositis. But how they are showing their actions, mechanism of actions of antimetabolites, they are particularly inhibit the S phase of the cell cycle, S phase, and how they are showing. These are the analogues. Pyrimidine, as you know, DNA contains various nucleotide bases in DNA. For DNAs, there are purine analogues are purine bases are adenine and guanine, and for pyrimidines are cytosines and thiamine. So these are cross-linked with each others. These are the nucleotide bases. So the drugs which are interferes, these pyrimidine analogues are interfere with this one, and purine analog interfere with this ones. So how they interferes? These pyrimidine analogues or purine analogues. These are the if I am talking about pyrimidine analog like 5-fluorouracil or cytarabine, they compete with, because these are the analogues, they have to compete with some endogenous molecule. They compete with deoxyuridine monophosphate, for the enzymes. That enzyme we call thymidylate synthetase, that is TS. So these competes with this one's compete with deoxyuridine monophosphate for thymidylate synthesis. Normally deoxyuridine monophosphates, these thymidylate synthesis will convert into thymidines, and then thymidine to DNA synthesis happens. So ultimately these interfere with this one, compete and interfere, and ultimately results in inhibitions of DNA synthesis. So they are showing their actions by this ways. These all like purine analog, pyrimidine analogs. These both molecules competes with their enzymes for these are interfere with the purines, and these are interfere with the pyrimidine. Another third one is folic acid analogs, and well-known folic acid analog is methotrexate. And I hope you all knows mechanism of actions of methotrexate. They inhibit the DHFR, dihydrofolate reductase. This is the enzymes which is involved in the synthesis of folic acids, right? So folic acid will not synthesize, and ultimately it will hamper the cell cycle. Right, so this is about the antimetabolites, pyrimidine analog, purine, and folic acid analogs. Next compounds are your in anticancers are natural compounds, and natural compounds are well-known compounds are in natural is vinca alkaloids, and in vinca alkaloids the drugs are vincristin, vinblastin. These are the well-known drugs. Vinblastins. Other natural compounds are texins, and in texins the drug is paclitaxel. Uh, docetaxels is another. Docetaxels. Another natural compounds are phylotoxins, and drugs are etoposides. So some enzymes are also used like L-asparaginase. So these are the natural compounds, and how they are showing actions. I hope you know these vincas or vincristin or vinblastins. These interfere or inhibits the polymerizations of microtubules. Polymerization of microtubules. Right, they interfere with the polymerization of microtubules, and these like your other molecules like amphotericins or some other molecule, they are interfere with the topoisomerase 4 enzymes. They like your this one L-asparaginase. This asparagine is required for the growth of cancer cells. So what this do, L-asparaginase inhibit this asparagine. So asparagine is not available for the cancers, for the growth of cancer. So this is about the natural compounds. Another compound which are used as a anti-cancers are, if you see, these agents are your hormones are also used. Some corticosteroids are also used. Corticosteroids like prednisolone, some anti-estrogens compounds like SCRM are also used in the treatment of breast cancer, selective estrogen receptor modulator. And under these drugs, there is a tamoxifen, raloxifene. These are the molecules which are used. Some for the others molecules like hormone, androgen, testosterone is also used. Anti-androgen is also used like flutamide. This is the anti-androgen flutamide. There are others drugs which are well known in the treatment of cancer is tyrosine kinase inhibitors. Tyrosine kinase is enzymes which helps in the growth of the cells, and tyrosine kinase inhibitors drugs are imatinib, gefitinib, erlotinib. So these are the drugs which are tyrosine kinase inhibitors are also used. There are some antibodies like rituximab are also used in the cancer. Antibodies like rituximab, trastuzumab, that we have covered in the immunomodulatory units also. So these are the antibodies are also used in the treatment of cancers. Arsenic compounds are also used. Some adenosine deaminase inhibitors are also used. So this is all about the anti-neoplastic agents. Next videos we will discuss about others compounds. Thank you.